Okay, Anil, I think we
All
are
set?
very
Nice.
live. Yeah.
Awesome. Cool. Then I guess welcome everyone to the Emmy seminar for March 12th. We're very happy today to have Erik Thiede from over in chemistry at Cornell talking about a variety of different methods used to recover molecular heterogeneity. So Erik, take it away.
All right. First question, everyone can hear me on Zoom? Any give you a quick thumbs up. Great, awesome. Okay. So my name's Erik. This is some work that I've been recently doing with my research group on sort of understanding not just what molecules look like, but how they can be different and how they're structured differently. And we've really been building some sort of tripod of simulation, electron microscopy, and machine learning to get at these things. So some brief acknowledgements. So of course, Flatiron Institute is where I started a lot of this work as a postdoc, and I carried it to Cornell. And then computational resources come from Flatiron Institute, San Diego Supercomputing, more recently Delta AI, and we've also most recent work has been supported by the NIH, and the slide needs to get updated. So to get an idea of the sort of things we care about, if you start looking at molecules, you quickly see that we draw these structures of molecules. Like on the left, this is a very big molecule, it's a protein. But actually, chemistry isn't just a single structure, it's a variety of structures that are all working together in concert. And you see this again and again and again, depending on what area of chemistry you're looking at. So in proteins, of course, AlphaFold was very famous for predicting the structure of proteins. But proteins are actually highly mobile objects. They are constantly interconverting, they're very floppy. And in fact, these floppiness, excuse me. So this floppiness is not just, it's not a bug, it's a feature. Often motion in the protein actually is how the protein accomplishes its biological tasks. However, at the same time, we see like different proteins can move in different ways. If you look at materials, the crucial issue in materials are materials defects. So very small scale defects can have outside impact on performance. These can be bubbles, dislocations, boundaries between domains. And the exact properties of exactly what dislocations occur, where they occur, can really change both the material properties and also the chemical properties of material. This is even taken to extreme, these idea of taking nanocatalysts, where if you have a catalyst, well, catalysis all happens on the surface of the catalyst. So the best way to maximize the use of your catalyst is to maximize the surface area by making it as small as possible. So if you have actually nanometer scale catalysts, that can drive up catalytic efficiency. But at this scale, the energies of catalysis are big enough that they can actually cause dynamic changes in the structure of the catalyst themselves. So we'll see these catalysts will grow, they will disappear, they will aggregate, they will constantly change behavior. But just to give you a scale of how small these problems are. So here I'm going to use this 200 nanometers here as my reference. So if I actually try and take these roughly at scale, you see that, well, this is my protein. This is about how small a protein is. It actually fits in one of these bubbles. My nanocatalysts are about this large. And over here, I have more or less the wavelength of violet light. So already, yes.
Quick question. Are you using your mouse to point somewhere? It's not showing up.
Well, okay. Do I have mouse pointy things on Linux? I don't think I do. Okay, okay. So at the bottom left, you have the wavelength of violet light. And what you can already see is that the phenomena that we're caring about is much smaller than we can see through an optical microscope. And we need new technologies, new ways to understand this. This has come, of course, the idea of using electrons. We know from quantum mechanics that electrons are heavier than photons. And heavier objects have a smaller quantum mechanical wavelength. So instead of focusing an optical beam with lenses, we are going to focus a beam of electrons with very powerful magnets. And okay, now we can really, because we're imaging these very small wavelength particles, we can start to see things at nanometer scales or sub-nanometer scales. There's a minor problem, which is we are bombarding a sample that is held together by electrons with electrons. And it's kind of like if you imagine imaging a building by throwing doors at it. You are going to destroy things quite quickly. Not only are you using doors, you're using doors moving at relativistic speeds. So as a consequence, we have this very interesting inverse problem. We have imaged something that is highly flexible, that is highly heterogeneous, and we have imaged it using an extremely destructive technique that results in huge amounts of noise. So yes, we have images, but we have images at incredibly low signal to noise ratios. And the sort of central computer science task here is, with us incredibly noisy data, how do we still recover something about the underlying chemical truth? So I'm going to talk about sort of two stories, one of which is focused on the biological world, where we've been trying to recover how conformational heterogeneity in proteins. We've been trying to understand how proteins move, how they change shape. And a key part of this is combining machine learning with physical models that allow us to actually recover this as well. And I'll also talk about very recent work on structured prediction tools. The second part of this, I'll talk about more material science applications, where we are using advanced electron imaging techniques and how we are starting this work and trying to understand how we can recover that data efficiently and understand complicated materials. So I mentioned that proteins are moving objects. Here's one concrete example. So on the left, I have shown HSP90. There's a cartoon representation of HSP90 in the middle. And HSP90 has this very famous hinge opening and closing. Specifically, now I will note, if you actually were to sit down and collect an actual movie, it wouldn't look anything like this. This is just an interpolation between linear open states. The actual motion would have a lot more thermal fluctuations, would be much jitterier, but it gets the point across. Now this opening and closing, however, is something that we know from biology. And in fact, we know what we can establish is that HSP90 acts like a little molecular pincer. By open and closing, it can affix to other biomolecules. And this is helpful at regulating how the cell operates. Now, one thing to note is there's a cycle here where, if I introduce ATP, the pincer closes. If ATP gets released, the pincer opens. If I can break this, I've actually created a drug. This is how galvanomycin works. Understanding these conformations are not only biologically interesting, it's also interesting from the perspective of drug design and also from the perspective of biotechnology. The more we understand about these motions, the more we can start to build almost like small molecular robots that convert energy into transport, convert energy into chemical synthesis. So in cryo-electron microscopy is a specific kind of electron microscopy that might give us a tool towards addressing this. What we do in cryo-electron microscopy is we take a solution with a protein, we freeze it in this very thin sheet of ice, and specifically like amorphous ice. Then we image this with the electron microscope. After we have done this, we collect these images, and you see one in the bottom right, which is all these individual copies of the protein. And then we go through and pick out all these images. Now, each one of these images is a very, very low signal to noise ratio. The signal to noise ratio is typically less than 1. It could be 0.1, it could be 0.01. And if you're going, it can be even lower than that. But the advantage is you have tens of thousands, hundreds of thousands, millions of these images. So traditionally, approaches have been largely based on sort of a signal averaging approach. If I average enough of these together, I can cut down the noise. And then I can recover what the electronic potential of a protein looks like. That's the second from the left at the bottom. And then using that, I can back out the positions of the atoms. The problem with signal averaging approaches kill all of the interesting motion that a protein has. It's great for getting a single structure. It's incredibly great for getting a single structure. It's so great for getting a single structure that was awarded the Nobel Prize in 2017. But you lose a lot of the heterogeneity. And I think nowadays there's this question of, okay, now how do we get that back? Now, this is, of course, a complicated problem. I'm seeing a thing in motion, and I'm seeing anything in motion that at very low noise. I might not even be able to tell at any one image exactly what state it's in, but maybe I can still solve the inverse problem. Maybe I can still get something about the motion back. So the idea we have here, and so to think about how do we connect protein motion to cryo-EM, is if we have a movie of a protein moving over here, every time we have frozen this, we've trapped it in a specific configuration. So you can almost imagine here if I have like a video of a horse moving, then I take a picture. On the right, we have all these freeze frames. And our task is to use these as to try and get these noisy freeze frames and recover the original motions. To make this more concrete, let's actually think about what is the mathematical object that is recovering. Well, traditional reconstruction tries to get a single structure X. Now, we're not trying to get a single structure. What I would say that we are attempting to recover is a probability density. We have or probability distribution over possible structures. We ask, say I take my hand and I reach into my solution, I pick one protein out. What is the chance that it occupies a specific problem X? So a lot of the literature in applied math here would call this a stochastic inverse problem. I have an unknown probability density that I have passed through a forward model, and I'm trying to recover it.
So you know what protein you're looking for, what the structure looks like, but you want the orientation now?
Oh, we don't. We know what protein we're looking for most of the time, because you prepared the sample, so you know. Okay, I put in 99% pure HSP90, but then I image it, and what I don't know is what the structure is. I might have some idea of what some possible structures would be, and different images will be in different structures. And the task is sort of not just recover all of these structures, but to actually recover their probabilities, to understand what is the chance that if I have a certain chemical condition, my hinge is mostly closed. If I then change the pH, if I introduce a drug, what is the chance that now it has opened up? And it's this sort of like probabilistic thinking that really is, if you've ever, this is sort of the core of statistical mechanics even, right? Where we have to, in physics, we have to use Boltzmann and thermal averages to understand how proteins behave.
So your punchline on the next slide was a little different than what I was expecting to see given the warm-up. It sounds like you're trying to just understand how likely is the protein to be in a particular state.
Yes.
I thought you were going to try to solve the puzzle of, like, I have this video, but all of the frames have been jumbled up in some random order, and I'm trying to reconstruct the sort of the most likely sort of sequencing of these motions from frame to frame.
So that's sort of, I would say, that's sort of almost like a next level analysis, right? You would say like, okay, now once I've established what these frames are,
somewhere where you'd like to go, where we could actually from these frames out of order figure out what the right order of sequencing is.
Yeah, but that is something that it is not clear if you can get this from this data alone, because this is a system at thermal equilibrium, it's frozen. When it's frozen, everything is just one snapshot of everybody in time. You could imagine, what you could imagine addressing this on the experimental side, though. What people do is time-resolved cryo-EM. So you do an experiment, and you say maybe you introduce some reagent or you introduce some change of the condition, and then at like time, 1 second to 5 seconds, a minute, you then freeze, and you freeze every time, and then over time you would actually see the changes in structure. The question is,
if I'm thinking about this like a video that I'm observing frames of, when you freeze here in time, I'm basically getting a very narrow range of times within this video.
You're getting
one time.
You're getting one time. And it is true. There's a little bit of fudge here, right? This video is clearly direction.
Yeah.
Right? The horse runs forward, it doesn't run backwards.
Yeah.
We know at equilibrium statistical mechanics that a protein at thermal equilibrium with a detailed balance, forwards and backwards are equally likely.
Right. So you really can only see the probability distribution within it.
Yeah, unless you start playing tricks with the experiment. where you start like collecting multiple different times, and then you actually maybe do want to say like, okay, how is my system moving over time? How is it responding to a change? Yeah, but so there are ways to do this, but here I'm only talking about the recovery problem. Part of the issue there is if you look at these images, right, they're incredibly noisy. And yeah, might be jumping ahead a little bit, but it's not clear that any one image unambiguously defines the confirmation state. So that also makes the dynamics recover very challenging. So, but we'll circle back to this actually when we talk about how we solve this problem. So the idea here, okay, we want to recover some true probability density over conformations. We call it different shape of a protein a conformation. And we're going to do what one does. You know, we introduce a model for the probability density, and then we are going to try and get our probability density to match the true. And we use a large amount of data. Of course, we can't compare these directly because we don't know the true probability density over structures. All we have instead are, this, okay, yeah. What we do, however, have are actual images. We don't have the structures themselves. The good news, however, is, well, we do have a good model for the imaging process. So even if we don't know the true probability density, if you sat down and gave me a model, the physics of the electron microscope is very well understood. I can sit down and I can model, I can sort of push forward this to a probability density over images. And that is the thing I can compare. So this is sort of the strategy we're going to pursue. From the experiment, we have imaged a probability density over structures. That is on the right. And that gives us a probability density over images. What we have is sample draws from that probability density. So we're going to replicate this process in silico. I heard that the word for this nowadays is a digital twin. So I
can still say in silico. It's okay.
Okay. Can I say simulation?
You can still say simulation. Just people won't think you're as cool as it.
Okay. Okay. Great. Okay. So yeah. So we set up a digital twin.
Yeah. He's really good.
Okay. And then we image the probability density. We have a simulated probability density over images, and this is something where we can compare, right? And this is just, you know, given them talking to a machine learning audience. Well, there has been extensive work on comparing two probability densities.
The image model is differentiable.
Yes, that's another point. The image model is differentiable. And I think you're maybe starting to see where this is going. Okay, so we're going to simulate, we're going to check how, and then we'll check our simulated images back match the experiment. And as you might guess, we're going to evaluate a loss function, back propagate all the way back to our model.
Yeah.
So how we're going to compare probability densities. We've done some work on this. We're just going to choose cross entropy for this work. We've been experimenting a little bit more with transport distances, energy distance, things like this. But cross entropy is already, you know, I'd say it's working well enough. And of course, it measures how close to probability densities are. It's minimized if they're the same. Of course,
this is just on pixels.
So the probability density is actually defined over images.
Yeah.
So then, yeah, but and we actually said write down this. It's going to boil down to I think it might be jumping ahead a little bit. It's going to boil down to like a function of, you know, the mean square error between images because we've assumed a Gaussian noise model.
Yeah.
And
how large are your images?
Depends on the experiment. I'd say around like 256 by 256.
And how do you know your forward model is really good?
Okay. Complicated answer. Answer number one, it's actually not. But it's good enough. Number two, there's been a lot of work, particularly to people like David Muller in the physics department and stuff, on really understanding the physics of electron microscopes. Gustav Kirkland also wrote a book on this. And within the various there's definitely approximations to how that works, such as, you know, the weak phase approximation. There's questions, okay, is the model...
I guess there are two levels of modeling. One is given, if you're talking about protein, given your, I don't know, base pair or whatever, how do you go from that to some structure? And then there is another level of how do you go from the structure to electron microscopy image, right?
Yeah.
And both of those are very non-trivial.
The second one, it's so
The second one in particular, I guess like if you do a takeography kind of thing, it becomes particularly non-trivial if you have big sample, multiple scattering.
Yeah.
Whatever.
but we don't.
Okay.
This is proteins they're largely carbon containing. They're all weak scatterers.
Oh, I see.
Weak phase approximation is
That's
good.
why protein people do TM a lot.
Exactly.
It's a really nice target.
Yeah, it's
I
nice.
guess
And the EM is
relatively
nice.
decent.
The signal to noise ratio is absolute trash compared. I was so bit of a
I guess
Bit
that
of a
the
digression.
standards are low.
exactly.
Oh.
I think that's the real answer to the question is like
Okay.
because all these problems are so complicated, our standards are much lower than you would if you were
Doing like crystal.
exactly.
Yeah.
Yeah.
Awesome.
And I got into this field looking at images like this. Quick digression. I'm talking to one of our postdocs who, so this is Alice Kennedy. She came from material side. She showed some really beautiful 4D STEM data,
Mm.
and I was like, "Oh wow, that's so clean." And she said, "Like, Erik, it's amazing that you think this is clean data." So I think the standards really are different.
Yeah.
Um,
Better of your expectation.
yeah. Yeah, and I think this is often okay. So coming back, cross entropy. You minimize the cross entropy.
So there are a lot of images, right? So
Mm.
then you're sampling over images.
Exactly right. We don't know this, so we're going to replace this with a sample
Okay.
mean. That's part. Why do we do this? We do it so we can replace this integral with the sample mean, and we don't have to know rho of y directly.
Okay.
We don't have to. That way, we don't have to do any density estimation on rho of y. Yeah, we've been playing around with it a little bit, and it's quite possible that it is an area to explore.
Seems reasonable to
Yeah.
do it.
Yeah, it's definitely things might work better, but we'll see how it goes. The imaging model, as this comes down to, it's a Gaussian imaging model where we assume Gaussian noise. If you're coming from the physics background, they typically assume Poisson noise. We're going to do Gaussian because most of the noise actually comes from diffraction from the ice.
Mm-hmm.
So there's a lot of background that sort of averages out to a Gaussian noise model.
So the
And-
reason why one does TM is because electrons are nicer than photons in some way, not because you can get a better resolution. I guess you can also do X-ray, but then you'll be doing maybe for crystals.
Yeah, I think so. The X-ray getting to like angstrom level wavelength, you have to use some really high energy X-rays, and it's much cheaper to. I mean, electron microscopes are extremely expensive,
Mm.
but they're a lot cheaper than building a
Synchrotron.
Yeah, so that's the that's the advantage there.
I'm just reading.
Okay. Yeah, we assume a Gaussian noise model. And then there's an imaging operator which contains all these approximations. That will be things such as, the weak phase approximation. In practice, what this boils down to is each protein exerts an electronic potential on its surrounding. And after you take a huge number of Taylor expansions, you find that the image is just a projection of the electronic potential in the plane. And that means that these images can be evaluated somewhat rapidly. I'll also note that there has been a lot of recent work on building differential libraries for this. One I'll particularly highlight is CryoJAX, which is one we've worked on. And that implements all of the imaging process in JAX in a object-oriented way using the Equinox library. It's really great. Uses all the Equinox tips and tricks. And that Equinox is a lot of, and CryoJAX has really powered a lot of the work that we've been able to do here. But yeah, it's really excellent work. If you ever want to get into this area, definitely check out CryoJAX.
Are you using TPUs?
TPUs? No. Do you have TPUs?
Not currently, but if you're doing cool projects in JAX, you should talk to Google because they like to get.
Okay.
They have TPUs and
Maybe I should do that.
they like publicity.
Okay. Yeah. Good.
I can give you some pointers on their grant.
Yeah.
Application process.
Yeah. That might be very useful.
I actually have another question about this.
Yes.
Is this sort of, are the structures really a continuum, or does this integral become a sum
So in
Or is it combinatorial?
it is really a continuum. Like in real life, it is a continuum, right?
Okay.
As with anything in practice, we can never, very rarely can we ever work with continuums, so we find some way to discretize. We're going to use Monte Carlo because this is a very high dimensional probability density. But in real life, yeah, proteins live in a continuum. There's definitely
Sure. So there's like a whole bunch of
focus.
degrees of freedom that are all scalar. Yeah.
Exactly. Things definitely center around specific, specific structures. And that's because, okay, we're gonna skip forward a little bit here, answer your question. So Boltzmann statistics is the probability of any structures proportionate to the e to the negative energy. E to the negative thing gets very large if that number gets low, and it gets low quickly. So things definitely cluster around specific structures often. Sometimes they don't, sometimes they do. And there's also really this notion that this is a very fractal landscape of this. It's not a very nice, the energy here is not a very nice smooth function. It's very, very, very rugged. And that often makes this quite challenging. It makes simulations of this also quite challenging. Yeah. Good questions?
I'm trying to visualize the algorithm, but I assume you're going to
Yeah.
show the algorithm at some point.
Yeah, the last thing we have to do is we have to, we need to introduce a model. And here there's multiple choices how you could do this. One of which is you could just discretize it using K-structures. And then you could optimize those structures. This is actually, you can do this, rederive a bunch of old algorithms this way. We've done some recent work on connecting this literature to gradient flows and sort of this these particle-based methods. Let's see if I can play this video. Yeah, so over here, this was a little toy model where we introduced this gradient flows that solve stochastic inverse problems under noisy conditions. However, other things you might want different things. And this is the question is we don't just want to know we don't just want a model of pi of x. Typically, we want a model of the structure for some downstream task. So for instance, two of those would be, I want a detailed physical model for downstream use, right? It's one thing to get a bunch of conformations, but what I really care about is predicting how does this protein now interact once I introduce a drug? How does it interact when I introduce different protein? How does it interact when I change the salt concentration? So in that sense, it's not just really we want to do is how can we actually use this information to build better physical models? The second thing I'll talk about very briefly, which we just started, is of course, how do we integrate this with generative neural networks? Because there's been a lot of recent work on generative neural networks for structure generation.
Do those work?
Sorry?
Do those work?
Kind of. I think you definitely can generate structures.
Yeah, but are they real?
My standards are low.
Yeah, my standards are low. I'd say they're very good at generating what I'd almost call protein rocks. So in general, they do a pretty good job. There's certain things they can't do. One of the things is they can't do flexible regions. And there's really a data limitation. Also, part of the reason we got into this is if you have a very flexible region for a protein, there are very few ways to characterize what that actually looks like in practice. And if you go back to this talk over here, in principle, if say I have a floppy loop or I have some tail that's wiggling in space, every time I took an image, that tail was pictured. That is not something that you get out of X-ray diffraction, because things like X-ray diffraction, X-ray crystallography, NMR, stacks, circular dichroism, they're all measuring all of the proteins at once. You get one measurement that encompasses everybody. So the measurement realities average out this heterogeneity. But here, because we're imaging single particles, in theory, we can get this data. And I think that's why we were interested that we could potentially, when you have something like AlphaFold, you could potentially get AlphaFold to do much better on flexible proteins. And this is also something people are noticing, that when you say these models work, well, they generate one structure. They often don't generate multiple structures. So there's a little bit of where they'll give you a good structure, but if there's multiple good structures, often they will not succeed. And I'll touch a little bit on this briefly when I discuss some very preliminary work.
Very, very hard problem.
Yeah, and I think part of it really was the data, right? And I think it still is the data. That's a little bit what we're trying to change here. Yeah, so I'm gonna focus on the middle here first. We're using a physical model. In the language of machine learning, this is an energy-based model for probability density. We do it because we have really, really, really good guesses for what the energy is, because we know a lot about the interactions and the physics of these proteins. So there's been a lot of work in putting together good models for these proteins. And the idea is, okay, we have noisy data. We want a lot of confirmations. This is an extremely hard task. So we're going to try and somehow put in a very strong physical prior. And the physical way we're going to do this is we're not going to start from scratch. We're going to use the delta method. We're going to introduce this probability density. Basically, these run, and this actually gets to your previous point about these trajectories. The way these work is they run dynamical equations to get motion over time. So if we are trying to connect to these simulations, one of the things we got into is like, okay, the simulation, the data itself doesn't give us any structural information. For that, we actually need some model of the motion. However, if we can modify this force field to better match our cryo-EM data, now we can go back, run a simulation, and now we're starting to maybe pick up some time information. That is a motivation. That is not we have shown that this is possible, but I think that maybe helps rationalize why we were pursuing that model. Okay, straightforward idea. We have a model for the energy. We're going to learn a correction to that, and we're not going to be fancy. We're going to do a 3-layer MLP, because if that's what you need, that's what you need. So you could imagine, yes, we'll do graphical networks, etc., but keep it simple to begin with. And then this learned correction will just adjust the energy. If I need more probability in one location, it'll lower the energy. But if I need more in another, it'll raise it. And then the idea is I take this, I can put that model right here, sample a bunch of structures, evaluate this integral with a Monte Carlo sum, and then that gives me a probability density that I can feed into my cross entropy. And then I just train everything all the way back, and then train it to get the parameter, and then update the parameters in the neural network. So here's a simulated experiment we did. One of the big issues with this area is, okay, great, I say I can claim these confirmations, but how do I even know that I've gotten the right answer? So on the left, we have a base model. This is a very rough and tumble molecular dynamics. It's designed to be cheap, fast, accurate, cheap, fast, less accurate. It's a coarse-grained simulation using the Martini force field. On the right, we have a very sort of detailed simulation that was much more slower. It was run by D.P. Shaw. And you'll notice we're already getting the physics qualitatively wrong. Over here, we have a protein. On the left, there is the protein folds into a little hairpin. And then there's this giant tail of disordered regions. On the right, our base model, this coarse-grained model, completely misses the hairpin state. And also, you'll notice the statistics of the tail are not correct. This tail should actually be pointing out a little bit more if I compare to the right. The question is, okay, I'm going to generate a bunch of simulated images using my reference. And then can I train a correction to this model to bring it back in line with my reference simulation? And that's the sort of simulated experiment that we set up for ourselves. So over here, we do this iteratively. So we train the model, rerun the simulation, continue training the model, rerun the simulation. This makes sure that our structure, because we have to reevaluate every step of gradient dynamics. But you'll notice as I progressively run this over time, I am able to actually fold this into this hairpin. This is something that Martini is not able to do by itself at the bottom right. And moreover, this tail actually does extend over, and it extends into larger RMSDs. So as a result, we are able to get, what's I think exciting to us is we're not just getting this folded structure right, we're getting we're improving the statistics of this disorder tail. And this is something that would have been completely impossible using signal averaging approach. You'll notice that a spurious minimum does occur around 3.5. That's because there's a few structures that form very similar cryo-EM images. But so basically, you could imagine that I form in the hairpin, it forms a little C. But you can imagine if it forms like a little U, it's very hard to tell the C and the U apart from each other, depending exactly how the connectivity. So many times you get very similar images, but the structures themselves could be a little bit different. So because there's inherent ambiguity in the images and at the noise levels we're working at, we do see the spurious minimum occur. But it still looks somewhat folded, and it's definitely better than where we started.
Between each iteration, you are running a simulation, and that simulation uses the reference.
It does not see the reference structures. It only sees cryo-EM images. So we run a simulation, and then we train the correction for a certain period of time using the cryo-EM images. Until convergence, then we rerun a new simulation and retrain the iteration. You can't go all in one shot because the beginning of the simulation is just far too different. So initial rating, when we just do one round of training the correction, we're going from 0 to 1. It gets better, but like we then need those new structures to actually be able to refine.
The simulation we're talking about here is the simulation of the electron microscope.
It is the simulation of that and also the simulation of the ensemble dynamics. So you have to run one of these, and then we simulate the image, and then all that data goes into a giant gradient descent loop where we optimize our correction. And then
I think there should be something. It seems like there should be some like
there should definitely be ways to accelerate a little bit and like reuse data from.
Because you're sort of like stopping the world and resampling. Where I feel like the machine learning kind of
would be to do it all together.
Approach should be to do something much more stochastic and noisy, where you're in the loop, sort of like taking 1 point sample. That's we could maybe talk about that.
We thought about that.
Oh.
The reason is that running these, so there's technical reasons. One is this trajectory. When you run this, because you're doing physical dynamics, individual points are extremely correlated with each other. So therefore, a lot of computational effort has to go into getting uncorrelated points with this. So to be able to do what you
Markov chain Monte Carlo.
It's a very it is the worst Markov chain Monte Carlo problem you've seen in your life. So you have to like run that. So once you invest effort into running that MCMC once, then you want to use that effort as long as you possibly can before you have to invest again. That said, there might be strategies where you take those points and continuously update them a little bit. That maybe connects some of this watch scheme flow stuff. That could be interesting. There's definitely ways this could be improved quite a bit. So I think this is not the most optimal training loop, and I would be very happy to talk about that point. Okay. Another thing, of course, is you could say like, look, this MCMC that is terrible. Why are you doing this? Why don't you just train a generative model? And well, that's something we're looking at as well. We started working this a few months ago. There are these tools such as AlphaFold 3 and Boltz. They generate structures. They're notoriously bad at generating ensembles of structures. They'll generate one good structure and then they'll keep saying that one structure. But we know from structural characterization that there's actual motion. These models, and that's not the fault of the models. It's that they were trained on large data sets that were built all using signal averaging techniques. So because there was not training data to be able to train on conformational ensembles, and I think this is one of the big issues when people are trying to build these models and now build generative models for heterogeneity. Where does the data come from? People are doing this using these simulations. These simulations are bad. Well, they're good, but they're definitely there. But there's a lot of errors in these simulations. So you're training on biased data. They're extremely expensive to do. And this is again, we're thinking, okay, maybe cryo-EM can come to the rescue here. It's a similar idea here. We take this generative model, we do it, we run it through a differentiable simulator of the imaging process, and then we train it to end. So we did this on a model system. This adenylate kinase that is an open and a closed state. These models generate only open configurations. And the question is, okay, we're going to build some simulated cryo-EM data set with the opening and the closed process. Can we recover both states as well as anything in between? We have some preliminary results here. It's just very standard. There's some minor tricks into getting the fine-tuning to work, which I won't bore you with. But we take this structure, we fine-tune our generative model, and then we are able to get a broader diversity of open and closed states. We're seeing how far we can push this. Of course,
you start with one of these protein folding models that tends to be kind of unimodal, and then you're fine-tuning it to
And where is the information for fine tuning coming from?
cryo-EM data, just giant bunch of cryo-EM data. Yeah. And then it's just we run it through the simulator every time, compare, calculate the probabilistic divergence, back propagate, and we keep updating the generative model.
So you're trying to get your generative model to produce structures that would end up matching diversity of TEM cryo-EM data. How well do they match with your forward model?
It is
quantifiable.
Well, I would say right now that is the calculation we're doing. This is like 2 months old. The first question is, can we even get any motion?
Ballpark of the fidelity? Like, are we talking about 99s, or are we talking about 60, 70, or are we talking about any sense? Because
it's also, you would have to, the question you would have to ask is, it's also a little protein dependent, right? Because, if you have a protein that's like an 80 to 20 relative probability, already that's a discrete model, right? So then, okay, how do you do this? I think I could give you a number for a Wasserstein distance, but I don't know how much that would tell you in isolation. And I don't know how much that would tell you if I then took this and then immediately went to another protein. And I think that's probably also because my intuition from talking to other people, the generative models, talked about fine-tuning these, where they were just trying to go from one structure to a different structure, rather than one structure to ensemble. They say some of these proteins work much better than some proteins are much easier to fine-tune than others.
That's believable.
Yeah. So I think it's going to be a little.
But from TM data, can you even? I don't know. TM data. Yeah. So are you're trying to match the distribution of TM images through the distribution of your substance?
We want to get the underlying distribution of structures behind the TM. I definitely feel like this is definitely one of those areas where even a little bit could go a long way. Just because it's
a kind of
it's
dividing by zero situation.
Yeah,
Go from zero to something. Yeah. Something is big.
Yeah. But also, we're also we definitely still have a lot of work right now, even making this fine-tuning protocol robust. Because if usual fine-tuning problems, if the learning rate is too aggressive, you stop folding the protein. You're running, these are big models to run, right? So you don't want it. Is generating 20 structures per iteration enough? Is generating 100 structures per iteration enough? One of these models can fill up a GPU, a single forward pass can fill up a GPU. Like a big GPU.
I realize maybe I don't understand this. So what you do is you take AlphaFold or something, and then you take your cryo-EM data, which you have processed through your discrete kind of clustering method, the first method, or
raw images. I've taken cryo-EM, I've picked out a bunch of protein images.
Okay.
Checked a bunch of structures.
Okay.
Compare them by running simulated experiments.
I think the statement is when you in your cryo-EM data, you see multiple images. Now, going from the cryo-EM data to the actual real structure is a difficult inverse problem by itself. But what what he's saying is that I know for sure there is a distribution because cryo-EM images have a distribution or there's a variety there. And your model only gave you one structure. So at minimum, you would like there to be, if I see 10 different images, I would like to see 10 different structures. That seems to be what they're aiming for. Am I right?
Well, yeah, I'd say we definitely would say like we have like biological characterization where we know there's motion.
Mm-hmm.
And it's so, you take this cryo-EM. Sometimes you will even run this and you'll do a bunch of signal averaging in the right way. And you're like, okay, I get two structures here that are different. There's probably a lot more in this data set. And then it really becomes this sort of ensemble matching where I want a button where I can press and I keep pressing that button and I keep getting new protein structures out that sample this ensemble. And then I want that sampling process to match my observed cryo-EM data.
So you take the AlphaFold network or another generative network, which produces a single structure.
Yeah. Press like 20 times.
Oh, okay.
Get a bunch of structures.
Oh, okay.
Then I
do
compare
the
with a giant
cross-entropy
batch of
loss.
cryo-EM, run the cross entropy loss. Yep.
Okay.
So yeah.
I'm sort of surprised that works at all, because if these models were trained on unimodal data to produce sort of unique responses, like trying to shovel in like distributional outputs and fine-tuning seems.
I think it's because you're not just printed on one structure, right? You're not,
Right,
these models are not just
generalized.
trained on one protein, they're trained on all proteins.
Yeah.
So as a result, there's probably, it's
So
not,
you're getting the multiple structures just from the stochasticity of the generated model.
Yes.
Oh, I see.
and you could also, one way other people have done, the way people did this originally was they would play games with the input sequence to get the model to produce different alternate structures.
Sure. You like.
Yeah, that was the
Corrupted
whole
it,
alphabet.
something, yeah.
That sounds like,
Yeah, so people would play those games.
Kind of.
So these models they are capable of generating like reasonable structures using what
Yeah.
they've learned from the rest of the protein burst.
Yeah, that's right.
Okay. Spatial heterogeneity materials. I've real brief. We also started wondering, we looked at this data and we're like, wow, there's so much more signal than everything we've ever looked at in our life. Do we have any way of working with that instead? So one of the nice things about this data is that you actually have a lot more interesting electron microscopy techniques you can do to it. Particularly if it's a crystal, you can go through, you can shine a little beam of electrons into the crystal and then watch it diffract. And that's a technique known as 4D STEM. And moving that beam across the sample gives you different diffraction patterns that tells you something about the local lattice orientation. My understanding is that most people, when they process their data, they do it by manual inspection, which is scary to me.
What are the 4 dimensions? So height, width, depth.
It is 2D imaging, and then at every location I get a 2D diffraction pattern. So it's 2 dimensions in real space and then 2 dimensions in Fourier space.
So when you shine the electron beam to a crystal, it scatters, and the scattered result gives a diffraction pattern like those dots in the red box. Those dots reflect the Fourier transform of the positions of atoms because of the way wave interacts with the arrangement of the crystals. So those dots are not the real position of atoms, but it's a Fourier transform of the position of atoms. That's a given one single diffraction. Now you take that beam, move it to another spot. You do that, you're going to get slightly different diffraction pattern, like those 2 boxes. So this 4D means you do diffraction, 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 diffraction. You move the beam position, that's the 2. And at each beam position, you get the 2D Fourier space, that's the other 2. So that's 2 times 2 is 4.
Okay. So we do, how hard is our stop at 4?
Okay. I'll here into questions about the
Okay, I'll just go really fast, just because there's an AI talk. In practice, people just simulate every possible one, compare this. We were like, you know what, let's just throw this through a feed-forward transformer and do the prediction immediately and it works well. Okay, that's that. So basically, instead of comparing to every possible diffraction found in a giant library, we create a transformer to do the prediction for us. It's faster than comparing to the giant library. Cool. Okay. I think this is, I don't know if it was you who, I think it said this at some point, where it's like 60% of problems in machine learning can just be solved by putting a feed forward. Yeah.
It's true. Yeah, it's true. I don't know if I said that, but whoever said that, I agree.
Yeah, it's. I mean, I don't think it's. I feel like 40% is linear regression. That's true. But then there's another 30% that's just like a encoder only or decoder only, and then like maybe there's another like tiny top of the bunch that needs new architecture.
Could you go back one slide?
Sure.
So I'm working on something that's template matching. What is the template matching?
You sit down, you say, this is what I think my system is. You download the structure, you simulate that structure from every possible structure from every possible orientation. And then you calculate a cross correlation. And then you pick the one that has the best cross correlation, which is also probably, okay, we also have bones to pick about this, but like, we think that cross correlation is actually very ambiguous and we should be doing probabilistic inference and blah, 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 blah. But that's a new project that is we started literally yesterday.
The cross correlation is slow.
It's relatively fast. There's just so many of these. There's because if you have 10 different phases, you have to do each one from like a few hundred orientations. And then that's just a lot of comparisons you have to do. And you might dip material thickness can also have an effect. So if you have, you have to now, okay, I've got 10 different phases. I have to do a bunch of orientations. I have to do a bunch of thicknesses.
The forward modeling is expensive.
Yeah.
And the space to forward model is practically infinite.
Yeah.
So if you really want to be really accurate, you'll be spending all your time doing that forward modeling. And you've got lots of forward models. You can do, the cross correlation is cheap.
Yeah.
But you just have lots of forward models to pick from.
In the forward model, we replaced this, like a neural network.
I into that. But it's a little bit like the MD situation. The space of possibility is literally really, really big, but the physical realities are only a fraction of that space of possibilities. So when you just do some like neural network that modeling that does not know about the constraints of physics, you waste a lot of effort in generating like modeling situations that are not meaningful.
Well, I guess I was suggesting to use the neural network to simulate the expensive forward model.
Right. So the expensive forward model essentially amounts to
So it depends on the level of complexity. For the stuff we're doing, so there's like sort of tiers of complexity. There's a cheap model, it's kinematic scattering. That is very, very fast. There's like, you can do block wave, which is slightly more complex. And then probably, I think what you were working with was the multi-slice. And that involves there you are solving the Schrodinger equation in one dimension. And that one, I don't know if someone has attempted to like, I think you might have issues that by the time you've trained it, I don't know how well you generalize. By the time, the things like, you have to be able to generalize because you could have also, if you have trained it, then you've also generated all the library patterns you can just match from. But I don't know if anyone's done it.
But this is something that I find like repeatedly. The lot of modeling situations in scientific domain often involves configurations in extremely large space of possibilities. But those configurations all very, very strictly obey the rules. So a fundamental challenge with neural network is that it's very hard to constrain neural networks. But the way how the scientists made progress is by hard coding, knowing those rules, and being able to just work with those possibilities that strictly obey the rules. So that's the tension, and I don't know how to make the best of the both worlds. But if there's a way, I think that can be transformative.
I guess I was hoping to waste an absolutely obscene amount of computation learning to.
Who gives you that?
I mean,
Google.
I mean, I think there
This is public.
I think so because we're trying to learn the inverse problems.
Yeah.
And I think that would be a thing, 'cause rather than learning the forward map faster, like I think learning the inverse map of the diffraction pattern to structure, then structure to diffraction pattern, potentially with some like conditioning on the input.
Yeah.
I think that's a place where it really could come up, because it's not just I think each multi-slice calculation is like manageable.
Okay.
It's not great, but it's manageable. It's when you have to start doing like a huge number of them, because you have to start considering a huge number of possible material structures.
But yeah, this is exactly
sweet
why.
spot is where the like full blown modeling is not possible, because once you're in the regime where full blown modeling is possible, people who are used to it have their standards very high, and they like, yes, I'm slow, but I get to this precision. Your thing is crappy, don't come to me, right? So you have to somehow get to the regime where the existing framework just cannot go. Then, then maybe, but then you have to still be able to verify. So that's also very difficult.
Or if you're in a situation like this where you don't care so much about specifically about the outputs of the system, but you're using it in an inner loop to solve some other problem.
Right.
And then it only needs to be good enough.
Yeah.
to guide
Yeah.
that outer
So then it's also a question of like, how do you, what is it? How do you define good enough?
Yeah.
Right? Because now it's going to have a huge impact in the part where you do care.
Yeah.
And
You
then
need some way to measure the
Yeah,
downstream
you need,
performance.
oh,
and then
that's always
see
the
whether,
verification is a challenge. But it's a very interesting question.
I'll also say for this specifically, I think there are opportunities, something when we visit a conference, and we can talk. I think there might also be an edge computing side to this, because people want to solve these problems on whatever shitty desktop is connected to their microscope. No offense to their desktops. I'm just, it's just like they aren't, and the desktop is like, so they don't have like desktops with like beefy GPUs hooked up to the microscopes directly. And I think there might be some, or even, I talked to them. I think part of it's sociological. It might also be, there might actually be like a noise part of it, because you just don't want like a giant thing that's causing a lot of vibrations. I don't know if that's true.
Oh, why don't they want to have GPUs?
It was,
You think it's
I had a long conversation with the salesman from Joel.
A quick
The next
question.
question.
Maybe we want to finish
Yeah.
streaming
Yeah.
and
Okay, yeah.
make
We
it
can
a
talk
private
about it.
discussion.
Okay.
Yeah. Okay.
Yeah.
Ciao. Please.
Maybe you should wrap up.
Yes.
Yeah.
Wrap up.
Wrap
Like,
up and- yeah,
conclude.
exactly.
Yeah, I think it's entirely that, like, it's just if you're going to spend $2,000 on a better microscope, and if you're going to spend $2,000 on a GPU attached to your microscope, it's just people will buy, the people who are buying this, they'll buy the microscope every time. And, I can't blame them, because you can do more things than that, but, So I think, yeah.
Maybe we come to a conclusion slide. Summary or,
Yeah, well, this is my group. They're really wonderful people. A lot of the neural network work done was done by Kawamura, who's sitting in the front. Jeffrey, who's the bottom left in the red shirt. Diego, who's in the middle in the white shirt, did the is still working on generative model work. Kwong on the left in the blue did the 4D STEM, and data was provided by Sungin Kim and Yao Yang in the chemistry department. Okay. And conclusions. Yeah, I think I'm just going to say it's a really interesting field, because you have this sort of integration of molecular modeling, this sort of like experimental data that I think has been underutilized and underexplored in this sort of, electron microscopy is mature, but I think there's still a lot that machine learning has to say in the space. And then, I think there's a lot. So yeah.
Awesome.